study the second type of tapetum, that is the secretory, also called parietal or granular type of tapetum. In this type of tapetum, its constituent cell wall remain in its original position throughout the microspore development. Substance are contributed to the anther sac by secretion from the inner face of the cell until their total breakdown at the time of pollen maturity. The secretory type of tapetum is more common among angiosperm and has been studied in far more detail than the amoeboid type of tapetum. Esklin and Godwin in 1968 have described in detail the events which take place in tapetal cell. In Helioborus foetidus, an example of secretory tapetum from the premeiotic stage to the stage of pollen maturation. At this stage, the sporogenous tissue, the tapetal cell, possesses mitochondria, plastids, and few spherical bodies, the pro-uvis bodies, and dictyosome with only a few peripheral associated vesicles. The cell wall are relatively thin at this stage and appear to be composed of middle lamella with a small amount of cellulosic primary wall. Before the onset of meiosis, the tapetal cell wall become thick and the cytoplasm appear more dense due to the increased number of ribosome and pro-uvis bodies. The thickening of the inner tangential wall of the tapetum, the wall facing the locule, are irregular. During meiosis, there is a further increase in the number of pro-uvis bodies and the size of the nucleus. The pro-uvis bodies, although distributed throughout the cytoplasm, appears to be more in the region of the cell nearest the anther cavity. At the tetra state, the number of ribosome further increase and that of the microtubule and plastid decrease. At this stage, two features of the tapetal cell deserved special mention. The pro-uvis bodies, which the number has further increased, now appear to be surrounded by a zone of ribosome which radiate from them like a spoke of a wheel. The limiting membrane of the pro-uvis body is discontinuous at those places which corresponds to the insertion of the ribosome vein. That B, the dictyosome, now appear to be associated with large number of vesicles at the periphery. The dictyosome and the vesicles are never seen associated with the pro-uvis bodies. The tapetal cell membrane at this stage appear highly convoluted. Soon after the microspore have separated from one another, ribosomes surrounding the pro-uvis bodies become prominent and the inner region of the cytoplasm contain abundant dictyosome. The pro-uvis body eventually pass through the tapetal cell membrane facing the locule without creating discontinuity in the later. Once the pro-uvis body have been extruded from the tapetal cytoplasm into the space between the membrane and the cell wall, they rapidly become coated with the sporopollenin and are now called uvis bodies. The inner wall of the tapetal cell now appear considerably thinner and a space appear between the wall and the cell membrane. Pro-uvis body continued to be formed within the tapetal cytoplasm and extruded from the cell and the sporopollenin is deposited on them. The deposition is initially on the side nearest the locule. All the tapetal cell wall have by this time disappeared and the cytoplasm has completely disorganized. The limiting membrane is ruptured and only a few recognizable organelles may be found in the residue. The uvis body are spheroidal structure, form only a few micron in diameter. They frequently fuse into a larger compound aggregate. It seems the uvis bodies are produced only by the granular tapetum and not by the amoeboid tapetum. The uvis bodies are involved in the external thickening of the exine, whose pattern is laid down by the spore cytoplasm in the tetra stage. Often the uvis body are capable of organizing exine pattern without any contribution for the spore itself. This suggests by the fact that in spite of degeneration of protoplasm in the pollen grain of Mirabilis jalapa, the exine pattern develops normally. Also in the male sterile Silenia pendula, normal exine culture develop. Thank you.